In the late 1920s, Scottish scientist Alexander Fleming made a remarkable discovery that would change the course of medicine forever. Fleming was working at St. Maria's Hospital in London, studying bacteria and searching for a cure for various infections. One day in September 1928, Fleming returned to his laboratory after a vacation. He noticed that some petri dishes containing Staphylococcus bacteria had been left out on the lab bench and had become contaminated with mold. As Fleming observed the dishes, he noticed something extraordinary: the bacteria surrounding the mold had started to die off. Intrigued by this observation, Fleming decided to investigate further. He identified the mold as a strain of Penicillium notatum and began experimenting with it. Fleming discovered that the mold released a substance that had powerful antibacterial properties. He named this substance penicillin. Fleming's discovery of penicillin was groundbreaking because it was the first effective antibiotic. Prior to this, there were limited treatment options for bacterial infections, and many people suffered and died from conditions that now could be easily treated. However, despite Fleming's groundbreaking discovery, it would take several more years before penicillin could be developed into a usable drug. It was not until the 1940s during World War II that the potential of penicillin as a life-saving antibiotic was fully realized. Scientists Howard Florey and Ernst Chain, along with their team at the University of Oxford, further studied and refined penicillin. They successfully conducted clinical trials and developed methods to produce the drug on a larger scale. The emergence of sulfa drugs' success sparked interest in the search for alternative agents. At Oxford University, Ernst Chain came across Alexander Fleming's article on penicillin from 1929 and suggested to his supervisor, Howard Florey, that they attempt to isolate the compound. George Dreyer, who had preceded Florey, had previously contacted Fleming in the 1930s to request a sample of his Penicillium strain for testing against bacteriophages, as a potential explanation for its antibacterial properties, which turned out to be unrelated. However, the strain had been preserved at Oxford. In 1939, Howard Florey formed a team that included Norman Hetley, an expert in fungi, who focused on cultivating Penicillium spp in large quantities, and Chain, who successfully purified penicillin from a mold extract. Florey oversaw the animal experiments. On 25 May 1939, the group injected 8 mice with a highly virulent strain of Streptococcus. Out of these, 4 were also injected with penicillin, while the other 4 remained untreated as controls. By the early morning of the next day, all the control mice had perished, while the treated mice remained alive. Chain referred to these results as nothing short of a miracle. The researchers published their findings in The Lancet in August 1940, detailing the production, purification, and experimental use of penicillin, which exhibited significant potency in protecting animals infected with Streptococcus pyogenes, Staphylococcus aureus, and Clostridium septique. In June 1941, Florey and Hetley made a trip to the United States. Due to concerns about the security of carrying a vial containing the precious Penicillium mold, Hetley proposed a precautionary measure. They decided to apply the Penicillium strain onto their coats to ensure its safety during the journey. Eventually, they reached Peoria, Illinois, where they intended to meet with Charles Thom, the leading mycologist at the U.S. Department of Agriculture, and Andrew Jackson Moyer, the director of the department's Northern Research Laboratory. Thom corrected the initial identification of Fleming's mold, which was initially thought to be P. rubrum but was later recognized as P. notatum. Obtaining a patent for penicillin was a contentious issue right from the start. Chain firmly believed in the importance of securing a patent, while Florey and others held the view that patents were unethical for a life-saving drug. This debate arose because penicillin posed a challenge to the fundamental concept of patents, as it was a natural substance produced by a living microorganism. In Great Britain, the prevailing belief at the time was that a patent could be granted for a process but not for the chemical itself. Both Merck in New York and Andrew Jackson Moyer filed patents for the process of penicillin production, and surprisingly, there was no opposition to these filings. However, When the war ended, British scientists found themselves in a situation where they would have to pay royalties for a discovery made within their own country. Interestingly, the production of penicillin at NG and SF, Northern Regional Research Laboratory and GlaxoSmithKline, turned out to be more than just a matter of historical significance. NG and SF had conducted their own research and development, utilizing their own mold culture, P. baculatum, and employing their own production methods. Consequently, they avoided any patent disputes, and their marketing efforts contributed to an increase in penicillin supply and a decrease in prices. By 1942, penicillin was being used to treat soldiers wounded in battle. Its effectiveness in combating infections proved to be a game-changer, saving countless lives and revolutionizing medical treatment. Penicillin became widely available for civilian use by the mid-1940s, transforming the field of medicine and marking the beginning of the antibiotic era. The remarkable impact of penicillin resulted in the Nobel Prize in Medicine and Physiology being awarded to Fleming, Chain, and Florey in 1945. Penicillin's isolation from other microorganisms gave rise to a new category known as antibiotics. Following similar methods of discovery and production, scientists in the 1940s and 1950s identified numerous other antibiotics such as streptomycin, chloramphenicol, erythromycin, vancomycin, and many more. Fleming, Florey, and Chain received the Nobel Prize in Physiology or Medicine in 1945 for their groundbreaking work on penicillin. Their discovery paved the way for the development of numerous other antibiotics, revolutionizing the treatment of bacterial infections and improving the health and well-being of people worldwide.